I will be talking about TrueAllele Computational Forensics for solving uninterpretable DNA mixtures. So, to tell you something about cybergenetics, we're an American company and we've innovated the accurate interpretation of forensic DNA evidence. Cybergenetics was founded in Pittsburgh, Pennsylvania, in the eastern US in 1994. In 1995, we solved the PCR stutter problem, which is important in DNA forensics. And in the year 2000, we eliminated the United Kingdom's DNA database backlog. And by 2001, over 20 years ago, we had solved the DNA mixture problem, which I will be talking about today with our TrueAllele technology. I'll also be telling you about how in 2005, we identified the World Trade Center remains, and in 2009, I testified in the world's first probabilistic genotyping case. In 2016, we did the first probabilistic genotyping DNA exoneration, and by 2020, we were validated on 10 unknown contributors to a mixture. Cybergenetics is reported on over 1,000 DNA cases, and it's used in over 90% of the 50 U.S. states. So back in 1995, we were working on genomics. And in computational genomics, we developed a way of rapidly building physical maps of chromosomes. And in essentially one summer, on a very limited budget, we built the first map of human chromosome 11 using a mathematical method that brought together different data that were low-quality data, but mathematically improved. In 1995, the same year, we overcame the short tandem repeat, or STR, genotyping bottleneck. There was a stutter problem of artifacts that appeared when the DNA was amplified with polymerase chain reaction, or PCR, and we worked out how to remove or overcome that stutter with mathematical models. In the year 2000, the British government asked cybergenetics to help eliminate their DNA database backlog. They were having human review of 350,000 cheek swabs a year with a staff that involved about 100 people manually reviewing in duplicate all of this data with an error rate of 1 in 2,000. So TrueAllele's computer automation cleared their backlog, and every year after the first year continued to process and interpret the data from DNA cheek swabs on 350,000 samples. The turnaround time was down to 5 days. Almost all the people were eliminated for a task that could be better done by computer, and essentially it was 2 or 3 computers doing the work of 100 people. Today I'll be talking about DNA mixtures. DNA mixtures are the most prevalent type of DNA evidence. A DNA mixture happens when 2 or more people combine their DNA in one evidence item. It can be 2 people or 3 people, like in a sexual assault, or 5 people, or 6 people, as in a handgun. We have reported on cases going up to 7 or 8 contributors, all in the same mixture. And the problem with mixtures is that they're hard for people to interpret. It's fundamentally a math problem, not a human intuition problem. The result is that millions of DNA mixture evidence items have been thrown out or ignored and not reported. And this still happens today. This is reporting language from a case cybergenetics just reported on last week. The lab looked at the data, and shown in red on the top is their conclusion from this vital DNA mixture evidence. Due to the data being uninterpretable, no comparison can be made to the reference sample. Well, that's true if you're not using TrueAllele. If you're trying to do it using lesser software or by eye, I guess it would be uninterpretable. It's like saying that a disease can't be diagnosed because you don't have a microscope. But if you do have a better DNA microscope like TrueAllele, the result that cybergenetics reported on the identical DNA data from this laboratory was that a match between the evidence and the suspect was 1,000,000,000 times more probable than coincidence. And so with TrueAllele, we move from the realm of absolute interpretation or uninterpretable with most complex mixtures giving no result and being uninterpretable to the realm of probability where information is preserved. So over 20 years ago, we published on how to make these uninterpretable DNA mixtures understood. We developed mathematical approaches to resolving these mixed DNA samples. The basic concept, which then went on to involve 10 years of development and a lot of math and computing, is that as you see in this picture, there's DNA peak height data in the center, and there's more than 2 peaks, which indicates a DNA mixture. But a computer can try adding together different genotypes, pairs of alleles. So in black, we have an 11-12, in white, we have a 10-12, and added together, we replicate the pattern. But to get probabilities, it's not enough to just get the best match. All genotype combinations and stutter effects and background noise and PCR variation have to be accounted for, and that's done using computing. The result is informative genotyping. We don't no longer have uninterpretable DNA mixtures. Sometimes there's little information, but the results are never inconclusive or uninterpretable. There just may be little information. What cybergenetics TrueAllele does is it unmixes the mixture data into genotypes, and then after the mixtures have been turned into genotypes up to probability for each contributor to the mixture, then the likelihood ratio match statistic provides a measure of information. In this case that we solved 10 years ago, there was a handgun, and there was a mixture of 4 people, and TrueAllele unmixed the mixture into 4 genotypes. 1 person was excluded, charges were dropped against him. 1 of the 4 contributors matched with a statistic of 400,000 ban units to an individual who then pled guilty. So by having science instead of uninterpretable mixtures, we had actual statistical results. 1 person was set free, another person was convicted based on science without even needing to go to court. How do we represent these separated genotypes? In this paper I've indicated at the top of the slide in the Journal of Forensic Sciences from 2011, we present the mathematics, which is Bayesian equations. Bayes in 1762 developed the initial mathematics of how evidence can update our belief or probability. So we may begin with some initial probability, like all the genotypes that have probability proportional to the human population, but after the computer examines the data, trying out 100,000 different possibilities of different parameters at every locus, different combinations and auxiliary variables, the result is genotype probability. At every locus, for each contributor, the identification information is preserved as a probability distribution. Of the 100 possible allele pairs for this contributor at this locus, there were, say, 5 where the probability was concentrated to reflect what the data was showing. This science is very old. It's based on probability from Laplace and Mendel from over 200 years ago. Bayes and Laplace, Mendel developed the genetic theory of how alleles work. And this approach of using genotype probability conforms with American standards issued by different standards organizations at the national level. Once we have separated genotypes, that's where most of the computation occurs. Now we need to quantify the information. But the power of separating genotypes is now each genotype corresponds to just 1 contributor. Inside the computer, when it solved the problem, It may have looked at 4 or 5 contributors together to explain the data. But when the problem's finished, it's produced a genotype, a probability distribution, for every contributor at every locus. It's separated into one contributor 4 times. So now the match statistics of the likelihood ratio are as simple as they are for single source DNA from one person. Each of those contributors can now be compared against an individual, a suspect or a victim, or an elimination sample to get a likelihood ratio. And it works just the same way as random match probability, the old method from 25 years ago for simple non-mixture data with one person. We ask, how much more does a suspect match the evidence than a random person? This gives us the likelihood ratio. At every possible allele pair, there would be a likelihood ratio, but when a comparison is made, say to this individual with a 12-14 allele pair, there's a ratio here of 98% probability to 3% prevalence in the population. That change in probability from 3% of a probability of a coincidental match up to what the data is telling us for this contributor of a probability of 98% for an evidence match is a ratio of 35. That's the likelihood ratio at this one locus. Multiplying those numbers together and across maybe 20 different short-tandem repeat loci gives us the full likelihood ratio. So there was a mass disaster in New York City, the World Trade Center attack on September 11th in 2001. There were 18,500 victim remains from the disaster site, and there were 2,700 missing people. Each of these elements is another missing person. And the task was to identify the victim remains, to take those 18,000 victim remains, which were often charred, small pebble-like molecules where ordinary DNA analysis by eye or simple computing would fail completely and give uninterpretable data, and compare them with DNA or genotypes from the missing people, from their personal effects, from genotypes reconstructed from their families, or both together. That was the DNA matching problem, to identify the victim remains. Cybergenetics was asked to help solve this problem, given the scope of the data and how impossible it was to solve this uninterpretable evidence. And to do so, we built our first TrueAllele supercomputer. It was a rack-based system. It was able to process all of that genotype data and make the matching comparisons. People worked on computers to enter the data and look at the results. It would take about 30 seconds to ask a question about an evidence item from the World Trade Center, and then the computer would do the rest of the work. Whether it would take minutes or hours, depending on how hard the problem was, that was all done on a computer server. It was fully automated, could use all the data. There are no data thresholds. That's handled by more powerful statistics that can model the data so thresholds aren't needed. There's no calibration needed, which was important for the World Trade Center because we were using data from 5 or 10 different kits from different laboratories. TrueAllele, when it solves a problem, gets all of that calibration information that other systems need directly from the evidence, the stutter information, the variance information. They're just hyperparameters in a Bayesian model that the computer figures out during its Markov chain Monte Carlo search. So a person would spend seconds asking a question, the computer would answer it, and then humans could review the data and we could issue reports. This is a smaller scale mass disaster. It was a bus crash that occurred in Komatipoort, South Africa. The police recovered burned victim remains, and they could identify many of the people, but they couldn't identify about 16 of the people, and so relatives were asked to submit their DNA to help identify the remains. Now, this was considered to be an impossible problem by the South African police. They had at most one relative for each missing person. And so they sent the data to us, expecting we wouldn't be able to find anything. But in fact, TrueAllele disaster victim identification using these relatives moved from what the lab called insufficient information to identified victims. So we were able to take the victim remains, here are the barcode numbers, and the samples taken from relatives, grandparents, sons, daughters, fathers, brothers, even where we didn't know the exact relationship, we had the computer solve it in different ways, and connect the inferred probabilistic genotype based on kinship from the family that represented up to probability the missing person who might have died in the crash to that victim remains and make the identifications. So how hard was this? Well, last year in Chandigarh, Cybernetics ran a workshop. And there were 20 students. At the beginning of the week, we introduced them to TrueAllele. The first day, they learned some of the concepts. The second day, they began working with the software. And for the third training day, we used this mass disaster as a problem that every student could solve. We set up one cloud database server that had 12 processors, and the task was to have each student use TrueAllele to identify the victim remains the same way Cybernetics had done it. How did that work? Well, the students succeeded. Everyone succeeded. What each student had to do was take the DNA data of 15 victim items, 16 family items, and the family references, and convert them into genotypes. And they were able to upload this data and have the computer automatically produce the genotypes. All the students had to do was to ask the question properly from the data, and the genotypes up to probability would emerge automatically from the computer. Each of the victim genotypes, compared with the 16 kinship genotypes from the family, produced hundreds of genotype comparisons, and across the 20 students, there are about 5,000 total match comparisons to calculate likelihood ratio, information statistics, the strength of association for each of these paired genotypes between the victim remains and the missing person. Before lunch, the students uploaded their data. During lunch, TrueAllele solved all the problems. The computer automatically produced the genotypes and automatically made the 4,800 comparisons. And after lunch, the students reviewed the identification results. That's the power of having a fully automated mathematical system where people ask questions and the computer does all the work of inferring the genotypes with probability, making the match comparisons, and presenting it in a form that people can understand. So the problem with manual mixture interpretation, there are many problems. Many authors, including myself, have written about this, but the problems are that it's inaccurate. It doesn't agree with the true information. In fact, in our courses, we've asked forensic scientists to predict how much information is there, and they agree with how computers solve problems, not with their manual methods. Manual mixture interpretation is subjective. The workflow introduces human bias. Manual mixture interpretation is a widespread. It's been used for millions of case items, but it only uses some of the data. It's very opaque depending on choices that people make, and it's been shown to be biased. It can only include or give no answer. It can't exclude innocent people, and most of the results on mixtures are inconclusive, and a case stays open. With TrueAllele computer technology, the approach is accurate. There have been over 40 validation studies, 8 published in peer-reviewed journals. The method is objective. There's no human bias. The workflow doesn't permit human bias. We're just asking mathematical questions from the data. It's widely accepted. Reports have been issued in over 47 of the 50 states in the US. It's used by laboratories. It's very transparent. The math is published. The software is available for people to test, including in any case, the opposition side, defense or prosecution, can test TrueAllele, and it's neutral. And the result is information. So how is TrueAllele used? It's used by prosecutors, defenders, police investigators, post-conviction. It's used in mass disasters for all sorts of different DNA, complex mixtures, touch DNA, paternity, and so on. I'll give some examples. Here's the first case ever done with TrueAllele. It's the first probabilistic genotyping case. And this was a homicide. A man was murdered. There was a 7% component of a mixture under his fingernails. And in the end, human review from the Federal Bureau of Investigation, the FBI, produced a statistic of 13,000. But TrueAllele was able to produce a number that was over 1 million times more. Why? It was the same DNA data, but much better DNA analysis, which led to information DNA results so the murderer could be convicted. This is a case, another homicide or murder, where a man was told not to visit his home again. His wife felt threatened, and the claim that his defense attorney made after his wife was murdered was that science could not distinguish mixtures that contained him or his children. But in fact, looking at light switches where his DNA was not supposed to be, because he wasn't supposed to be present, the computer could figure out that the murderer was present, but his children weren't, and on other items, his children were present, but he wasn't. So TrueAllele can resolve mixtures of relatives. This is a serial rapist in Pennsylvania, and here there were uninterpretable results by human review, and after we examined some of the evidence from clothing from the female students who'd been raped in their apartments, this uninterpretable data produced a match statistic of a quadrillion. That's a 1 followed by 15 zeros. There's a lot of information in this uninterpretable data that would actually was very informative. This was another rape case. This was a father-daughter incest rape where, because of the relatives, the mixture was uninterpretable, as the crime lab said. But in fact, TrueAllele produced match statistics of over 1 million connecting the father to his daughter's clothing that led to his conviction. In Maryland, in Baltimore, Maryland, there was a serial rapist who had not been convicted after 3 or 4 cases because he could always tell a story. In this case, it was hard to get past the story that TrueAllele found the defendant, the victim, and her boyfriend all on the same item, on several items, clothing, a phone, a belt, and there was no story he could tell to get around the information from the DNA that TrueAllele found, and he was convicted. TrueAllele is also used to exonerate innocent people. We've done over 10 exonerations, helping to free people from prison or clear their name. This is Indiana versus Daryl Pinkins was the first case we had done. Daryl Pinkins was wrongfully convicted for a sexual assault he was not involved in any way. He was imprisoned for 25 years. TrueAllele was able to prove that he wasn't there and identify 5 genotypes to satisfy the court, and he was released from prison. So the point is that instead of viewing DNA analysis as producing data and then looking at it by eye or with simplistic software, often getting an uninterpretable or inconclusive result, with TrueAllele and mathematics and computers, DNA, even the most complex DNA, is an information science. And what TrueAllele computing provides is information and automation. With information, there are no inconclusives. There may be little information, but a result can always be given because the computer can accurately weigh the DNA information. The automation frees up people. The computer can do the work without needing to hire hundreds or thousands or tens of thousands of people to look at data. The interpretation is automated, and it's very easy to use. The users, forensic scientists, ask questions in seconds, and then they get answers. There have been 8 peer-reviewed validation studies starting in 2009 and proceeding through the year 2020. These validation studies have been done on low-level DNA, maybe 1% components, as well as large amounts of DNA, from 2-person mixtures to 10-person mixtures. And what we find is in these studies, the TrueAllele results are accurate. They're reproducible. You see the blue and the green bars are giving the same information numbers across these different cases when the computers run twice, and more informative than human review, shown in orange, in this case by a factor of 6 log units of likelihood ratio, or 1,000,000. So it's more informative. Also, in this paper published 10 years ago in the Journal of Forensic Sciences, the blue cloud is representing the information for 85 DNA matches in cases looked at by the New York State Police. But they've produced a result in only 30% of the cases. So this study showed that even on simple DNA mixtures of 2 and 3 people, the labs are throwing away 70% of the match information, calling the data inconclusive or uninterpretable. The final result that we've produced in terms of validation study has gone with studies from 5 unknown contributors, and in 2020 we published this paper with 10 unknown contributors. And the computer produced results. We processed them at Cybergenetics. The Cleveland Crime Lab ran them independently. We got the same answers, and we were able to solve 10-person mixtures. So thank you very much. I appreciate your staying. If you'd like more information, please visit our website, cybgen.com, or contact me, Dr. Mark Perlin, and I'd be happy to talk with you more about DNA analysis as an information science that gets rid of uninterpretable evidence and turns the same data into useful information. Thank you very much.